we consult to Wikipedia for a review of the Bohr effect and also the Haldane effect, which provides an explanation for how oxygen and carbon dioxide and carbon monoxide binds to the hemoglobin. The Bohr effect, as we've seen in the class, there's a sigmoidal curve which shows the oxygen's affinity and the binding of oxygen to hemoglobin. It is called oxygen hemoglobin dissociation curve. Okay, we can click this one as well. Structure of hemoglobin, sigmoidal shape. Here we have the oxyhemoglobin, also called percent saturation, and which is the hemoglobin that is bound to oxygen. As a percentage, it's 100. And here we have the oxygen's partial oxygen pressure. And we have the sigmoidal curve with decreased pH or increased temperature, or with the presence of DPG, Ch03 DPG. We've seen this in the class. The curve shifts to the right, and as a Bohr effect, with the carbon dioxide's concentration increases, the pH is lowered, and in vice versa, when DPG is not present or pH is higher, then the curve shifts to left. This is myoglobin's curve. This is pure hemoglobin, and this is the actual with DPG bound hemoglobin. This is the shape of the hemoglobin, so that's it. This is called Bohr effect because it explains the affinity to oxygen and binding of the carbon dioxide instead of oxygen when the oxygen levels drop, and then carbon dioxide gets to bind to hemoglobin. And in tissues, because the oxygen concentration is low, then the hemoglobin releases the oxygen to the tissues. We have this carbon dioxide. So carbon dioxide can be carried by itself as a proton, or this one bicarbonate ion here. So we've seen the T-state stabilization, T-state and R-state, the tense state and the relaxed state. In the relaxed state can be achieved by cooperative binding. And carbon monoxide is a competitive inhibitor for oxygen. It binds to the same place as oxygen, and it is irreversible. When it is bound, it's called carboxyhemoglobin. Here you can see the shape of carboxyhemoglobin, which carbon monoxide is bound to the iron, central iron. Hemoglobin's affinity for carbon dioxide is like 200 times stronger, so it becomes toxic. And then we have the Haldane effect. And the Haldane effect, hemoglobin's ability to carry carbon dioxide in the deoxygenated state. So when the oxygen is released, carbon dioxide can bind to it. So carbon dioxide is carried in blood in three forms: dissolved gas, dissociated carbonic acid, or bound to proteins in the form of carbamino compounds. So it can diffuse as a gas itself as well. It can be carried as this ion. And also, as I tried to remember in the class, it binds to the hemoglobin as carbon dioxide as well. So we should have a picture of it. Carbamino compound. Is that? No. Yeah, yeah. A very nice description. This is the hemoglobin. It has the amino groups. Carbon dioxide binds to these amino groups. This is called carbaminohemoglobin. It doesn't bind to the iron, but it binds to the amino groups. It's a different forms a different structure. In the tissues, we have this carbon dioxide getting out, oxygen coming in, and the red blood cells, we have this carbaminohemoglobin formation and also the bicarbonate ion formation. And also in the plasma, we have the dissolved carbon dioxide, and in alveoli, which are present in the lungs, we have this oxygen leaving alveoli. So we thank Wikipedia for this clarification. Okay, so oxyhemoglobin dissociation curve. We've seen this in the class, but again, there are some better visualizations of it. This is the normal curve, dissociation curve. It's the right shift. Here is the left shift. Again, the oxygen partial pressure and hemoglobin saturation. It says to the left shift, we have oxygen affinity increase and unloading of the oxygen decrease, which is caused by increased pH again. Fetal hemoglobin, carbon monoxide by bond hemoglobin, and the right shift is the opposite of it. Oxygen binding decreased and the unloading increased. Now, because we are talking about the fetal hemoglobin, so we have in the PDB, I realized that there is a fetal hemoglobin page as well. This is the fetal hemoglobin. This is the normal one, alpha and beta, 2 alpha chains and 2 beta chains. In the fetal hemoglobin, we have 2 gamma chains instead of beta chains. 2 subunits make one big difference. So fetal hemoglobin has a higher oxygen affinity than adult hemoglobin under normal healthy conditions. Here, one important difference in fetal hemoglobin, the 2,3-BPG molecule does not bind to the fetal hemoglobin. This is the normal hemoglobin. Let me see. 2,3-BPG are shown in purple. It says. And the neighboring amino acids are shown here. We have the heme group separately in here, 2,3-BPG in here, and we can change it to fetal hemoglobin, which doesn't have the 2,3-DPG. As you might remember, 2,3-BPG shifts this curve to the right, but in fetal hemoglobin, we need to shift it to left. So that's why it doesn't use that molecule. We can show it the entire protein. Here is the fetal hemoglobin and the adult hemoglobin. Heme group, the different amino acids bound by this DPG. Obviously, everything that changes the affinity or binding capacity, has a ability to change the conformation, and by changing conformation you change the functionality of that protein as well. Here it shows the picture of it, and the amino acids are marked. This is histidine, again histidine. They are forming the bonds with 2,3-DPG, but in the fetal hemoglobin we have different amino acids. Different amino acids are located. So probably this is the only difference between the beta chain and the gamma chain that's present in the babies.